Good morning, students. As we are entering the day 6 of our BIIS-11 workshop, today we have with us one among us, BIIS-11 student, Ms. Kirti Saxena from SRM CET College, Bereli, is here with us to present a lecture on clinical trials. I invite Kirti Saxena to deliver that talk.
Thank you so much, Professor. I'll be sharing my screen with all of you. Is it visible?
Yes, ma'am.
Yes.
Should I start, Professor?
Yes, but your screen is flickering, I think.
Is it fine now?
Go to the full screen mode.
Now is it fine?
No, your screen is very flickering.
Now is it visible? Now is it visible, Professor?
Hello.
Hello.
Yes, you can start.
Okay, good morning, all of you. My name is Kirti Saxena and I am a student of B.Pharm final year from SRM CET Bareilly. Today I am going to present this short talk on clinical trials. So starting with our content, that is, at the end of this, we will be able to know about a short introduction of clinical trials, what is the drug development process, the preclinical trials, the various phases of clinical trials, and the regulatory approvals involved in this. So starting with clinical trials, what clinical trials are? Clinical trials are research studies performed in human subjects that are aimed at evaluating a medical or surgical or behavioral intervention. Whatever we create novel and we have to publicize it in market, initially we have to perform clinical trials. And the word clinical trials itself signifies that the trial will be performed on human subjects. They are the methods by which researchers find out if a new treatment, like a novel drug, diet procedure, either a nutraceutical diet or some changes in the normal diet that are used for the CKD patients or some diabetic patients, or any medical device, for example, pacemakers, either it is safe or effective in people or not. A clinical trial is used to learn if the new treatment is either more effective than the existing treatment or is having lesser side effects. Because whatever we are creating new, our aim is to provide better safety and efficacy to the patients. Whenever the regulatory trial commences, they initially need approval from the regulatory authorities to begin. The scientists perform various kind of laboratory tests that are biopharmaceutical studies, and then test the drug or the intervention on animals for checking its potency, its safety and efficacy. Then only the trial commences. Initially, when a compound or an intervention, when it gives promising results in the preclinical and laboratory test, then only they are carried forward for the clinical trials. The clinical trials are conducted only when initially a satisfactory set of information is gathered from the non-clinical data.
Your screen is flickering too much.
Is it fine now?
No, it is flickering. Keep on flickering the screens.
Will I reshare this?
Yes, yes.
Wait, Professor, I'll be resharing it. Is it visible now?
It is visible, but still it is flickering.
I'm not sure of the reason.
From your side, it is clear?
Yes, sir, from my side it is clear.
But the screen is keep on flickering too much.
If possible, you will be sharing this presentation?
Okay, then I will share this.
Yes, that will be more convenient, I think. Yes, Professor, it is visible now.
Yeah. Yes, you can continue.
I think we are on slide number 5.
5. This one, right?
Next one, this one. Thank you, Professor. Then should I start?
Yes, continue.
So starting with the drug development process, the process is majorly divided into 3 steps. Initially, the drug discovery, that is the lead identification, lead optimization. Then the preclinical development, in which we perform the safety testing in animal subjects. Preclinical word signifies animal studies. And then the final and determining clinical trials. These 3 are the major steps whenever we are developing a new drug. Next. This is a flow chart that is depicting the whole drug development process, starting from the preclinical data, then IND filing, the various phases of clinical trials, then NDA approvals, and then the phase 4 of clinical trials. I'll be explaining it better with the next pictorial diagram. Next slide, please. So here is a pictorial diagram which is representing the whole drug development process. Drug development is a very long process, as any new molecule takes almost 25 to 30 years to get published in the market. It is very easy to have drugs in market, as we see from a far out view. But being a pharmaceutical professional, I know that this process takes number of years and a lot of patience to have a new drug molecule published in the market. Starting from the preclinical analysis, in which we test our drugs on animals. We use animals, small and large, both kinds, and when they give us promising results, then only we apply for the IND application, that is investigational new drug application, which is filed with FDA and takes 30 days of time. In 30 days, the FDA review your application, and if they find it promising, then they allow you to continue with clinical trials. And if it is not sufficient, then they'll ask you for adding more information, or If you are not able to provide that much promising information, they will reject your application. After the approval of IND from FDA, the phase 1, 2, and 3 trials are continued. They take almost 10 to 15 years because it is a very, very long process. After these 3 phases, the NDA review is done, which take a time of ranging months to years. It takes almost 7 to 8 years for a drug to get NDA review. And then only after the NDA review, the drug get marketed. And after even marketing the drug, the four trials of the clinical process continues, which is the post-marketing surveillance, in which we follow up the patients to which the drug is being sold and continue to have the data from the patients who are using it. Next slide. Generally, we consider only 4 phases of the clinical trial, but even phase 0 is a very important part of the clinical trials, which is not counted in many processes. The phase 0, 1, and 2 are exploratory kind of trials in which we develop the proof of concept that our drug, which we are drug intervention or molecule, which we are claiming to have some effect. We develop the proof of concept in these 3 phases. In the later on 2 phases, the phase 3 and phase 4, these are pivotal. These are very crucial for the information, and they are the determining part of the clinical trials. Next slide. Before a sponsor can initiate testing in human beings, extensive preclinical and laboratory research is required because you can't test any drug intervention or molecule in human subjects likewise. You have to be very, very efficient and very, very promising in preclinical and laboratory tests. Then only you can go for the human trials. This research involves number of years and experiments on humans and animals both. If the animal stage of the trial is successful, and then only the FDA is requested for the IND, and if they approve you from the IND, then only you can continue your trials in human subjects. Next slide. So what preclinical studies are? The preclinical studies, the word preclinical itself signifies that we are going to experiment our drug, intervention, or molecule in animals. It is the research using animals to find out if a drug, procedure, or treatment is likely to be useful. They take place before any human testing is done. Next slide. There are several kinds of animals that are used in preclinical trials. Generally, they are classified into 2 major groups. One is the small animals, and second is the large animals. The animal trials are totally governed by the CPCSEA, that is Committee
Kirti? Kirti? Kirti, are you there?
Hello, am I audible?
Yes, Kirti. Yes, you can continue.
Am I audible, Professor?
Yes, Kirti, you can continue.
Sorry for the glitch. That was a power cut. So starting with, it is about small animals that we use in clinical trials. Rat, mice, hamsters, rabbit, guinea pigs, frogs. Each animal is having its own specific characteristics. For example, rats can't be used for emetic and anti-emetic drugs because they don't have vomiting centers in them. Their innate physiology is specified for that. And that is a boon so that we can use anti-cancer drugs which can be tested in rats so that they don't show emetic effects. Mice are a kind of very small and very fast animals. Each college or the organization which is having animal trial permission, they have to be licensed from CPCSEA, that is Committee for the Purpose of Control of Experiments on Animals. Hamsters, rabbits, guinea pigs. Guinea pigs are very sensitive for antihistaminic drugs, while rabbits are generally used in pyrogen testing for sterile preparations. Next slide. Then these are some large animals: dogs, chimpanzees, monkeys, sheep. Sheep are generally used for asthmatic drugs and the drugs which are used in hemophilia. They are very prone for that type of preclinical studies. Whenever we talk about preclinical studies, that are the testing in animals, either small or large. Next. It is a small exemplary pictorial view that how preclinical trials goes. That, for example, you tested a molecule in vitro, then you have promising results, then you go for the preclinical test, you check it in your rat, mice, or whatever small animal. You study the mechanism of action of that drug, molecule, or intervention, that how it is acting in your body. You perform the toxicology test, potency assays, consistency assays. Then you apply for IND filing, in which you submit all these data. When the FDA or the governing body found it considerable, next slide. Am I audible? Then only the review is done by the Central Drug Research and Evaluation Center. They review your protocol, and then if they found it promising, then they continue the testings. Preclinical studies can be designed to identify a lead candidate from several hits. Whenever we decide or design a new molecule, we have number of experiments and number of subjects. We have variety of drugs for a same treatment, but we have to optimize that. We can't take 100 or 1,000 of molecules for trials. We have to optimize the bestest one with least possible side effects and maximum efficacy that we can continue in our preclinical and clinical trials. Even preclinical trials are governed by the CPCSEA and they have several limitations. You can't kill or sacrifice an animal without any considerable purpose. Because as much as humans are important, their life is worthy, the life of animals and the cruelty against animals is also regulated. We have to develop the best procedure for the new drug scale up. We have to select the bestest formulation because variety of formulation, the kind of formulation also defines the efficacy and safety of a drug. For example, you cannot give oral formulation of a drug which get destroyed by your enzymes from the gastric mucosa, from your mouth. We have to specify. For example, proteins, hormones, they can't be given orally. They have to be administered ion, intramuscular or intravenously. We have to determine the route, either it is oral, intravenous, intramuscular, or the frequency of dosing, and the duration of exposure, because every drug is having its own individual half-life. We have to identify for which frequency and for how much duration we can leave our animal to be exposed with the drug. And ultimately, these all results, the data provided from all these tests, it supports the intended clinical trial design. Whatever we get as an outcome from all this test, that is the supporting document for providing a proper and successful clinical trial design. Next slide. The toxicology and safety studies are identifying the potential target organs for ADR and defining the therapeutic index. ADR is the adverse effect, the harmful effect that we obtain from a drug at its normal therapeutic dose. Overdose, underdose is not considered as ADR. For example, PCM is having 2400 mg per day as its standard dose, which means that you can administer 2400 milligram of PCM to a patient having pyrexia, that is fever, in a set of 600 mg every 4 hours. You can't give more than the daily prescribed dose to any patient because that may prove fatal or very much toxic for the target organs. Likewise, PCM is also indicated for its harmful effect on livers as a hepatotoxic agent. Pivotal preclinical safety guidelines generally require regulatory oversights. The regulatory oversights are defined by US FDA, that is United States Food and Drug Administration. It is a global center and regulatory agency, and the protocols they have designed are very, very efficient. So whenever we plan for a molecular trial, we always go for the FDA guidelines. The good laboratory practice. The animals that you use in trials are practiced in laboratories, and it is very, very efficient to have a knowledge of GLP so that you will be able to handle animals. You know how to dispose the waste of. But even if you are dissecting an animal, there are several protocols that how you will be opening the animal, how you will be discarding the remainings of that animal, what organ you will use, how you will obtain it. Everything is specified in these procedures. International guidelines, including the ICH guidelines, that is QSEM, Quality, Safety, Efficacy, and Multidisciplinary Guidelines, that are mainly used. ICH guidelines are also very, very essential sort of guidelines that clinical trials and preclinical studies follow. Next slide. Pre-phase 1 activities. Whenever we are about to start the phase 1 activities, that is beginning of the clinical trials, we have to determine some of the pre-phase 1 activities, that is initially preclinical data review. Whatever the data we obtain from the animal and laboratory trials, we have to document it up regarding the safety, efficacy, toxicology, and ADME. ADME is pharmacokinetics, that is absorption, distribution, metabolism, and excretion of a drug. Then the second part is the preparation of an investigator's brochure. IB is a very important document in clinical trials, and it is a summary of the preclinical data that we have obtained from the trials and its clinical extrapolation. We are having the data that promises the effect in animals, but we have to extrapolate it to have an assumption about how it will react in the human subject. The investigation brochure also includes the prediction of clinical effects and the safety. And third, and the most important part, that is filing of investigation new drug approval with DCGI. Globally, US FDA is the governing body. In India, CDSCO, the Central Drug Research Organization, is the governing body, and the Director General of CDSCO, that is DCGI, Drug Controller General of India. He is the one licensing authority in India who permits you for clinical trials. Next slide. So we have completed with the preclinical studies. That is, we have promising data from our animal studies. Then we inculcate all that data. We documented it with proper protocols, and then we apply for the IND filing, that is investigation new drug application. This is filed with the FDA. Every country is having their own FDA, and globally we have the US FDA. Once preclinical studies have indicated the safety and efficacy of a drug, it has to be filed with the regulatory authorities. For obtaining the approval for phase 1, 2, and 3, always remember that IND filing approves you for only phase 1, 2, and 3 of the clinical trial. Phase 4 is done only and only when you have promising results from phase 3. The results are reviewed by the NDA, and then only when you get NDA approval, you will go for the phase 4 trials, because phase 4 includes marketing. We will discuss it further. The major content of IND application is mentioned here, that I have told you, that the preclinical data, all data that we have from animal studies, information on the composition and source of a drug, from where you have obtained it, either it is a synthetic molecule, a herbal drug, a chemical extract, or whatever. The manufacturing and chemical information about the chemical formula and the manufacturing procedure which you are using for the drug, the proposed clinical plans and protocols, and animal ethics committee clearance. Ethics committee clearance is very, very important, because until and unless you are not having an ethics committee clearance certificate, you can't go for animal or human trials. Next slide. So starting here with the phases of clinical trials. According to US FDA, clinical research refers to the studies or trials that are done in people. As I told you, that clinical trials are always considered when we try a molecule or intervention in human subjects. It includes 5 phases. That initial is phase 0, that is called as microdosing trials. Then phase 1, 2, and 3, which we do after IND approval. And after this, phase 4, 3, we apply for NDA approval. And if we're getting it, then we'll go for the phase 4 trials. Next slide. It is a summary of all the phases of the trials. The time, approximate time that each take, the number of participants each involved, what are the study purposes, and number of drugs that go for the trials. Here 70%, 30%, and 25% is the significance that, for example, if we are having 100 molecules for a same effect, then they'll go further and get reduced in each and every phase. Because not all molecules get their effect, and not all molecules are about to be published. If we are having 100, only 70 will go for the phase 1, only 33% of that will go to the phase 2, and then 25% will go and remain stagnant up to phase 3. And at the end, we are having only 1 and 2 molecules that are published for the post-marketing surveillance. Next slide, please. So starting with phase 0 clinical trials, as I told you, it will also called as microdosing studies, and that is first in human trials. For the first time, human subjects are introduced with the molecule under intervention that we have developed. Here, we are having a single subtherapeutic dose. What a subtherapeutic dose is, for example, 50 mg of a drug is defined as a therapeutic dose, which is needed for having its effect. Then a dose which is lesser than 50 mg is called as subtherapeutic dose. And it is given to 10 to 15 subjects to yield pharmacokinetic data, that is the absorption, distribution, metabolism, and excretion kinetics, that how the drug is behaving in the body, and imaging specific targets without introducing pharmacological effects. Every drug, the human If we talk about the human physiology, every human is having different physiology. Even for the same kind of molecule, if we are testing it in 15 individuals, each and every one is about to react differently. Pharmaceutical companies perform phase 0 to decide whether the drug candidate is best pharmacokinetic parameters in humans. If a drug is having lethal or fatal effects in phase 0 trials, then it is withdrawn and it is not continued further for phase 1, 2, 3 trials. Next slide. What is a microdose? As we have mentioned that phase 0 is a microdosing study, it is less than 1 by hundredth of a dose of a test substance calculated to produce pharmacological effect with a maximum dose ranging equal to or lesser than 100 micrograms. Can you imagine 100 micrograms, such a small amount? Whatever dose we are having for the therapeutic effect, for example, 600 mg of PCM is needed to have the antipyretic effect, that is, it will reduce fever. Then, when we are having the microdose trial, we will reduce its dose in the proportion 1 up to 100. This much reduction in dose is considered as a microdose. Next slide. The major aims of phase 0 trials is to aim and find out that either the drug reach the disease cell, because whenever we have or consume a drug, it is not 100% reaching your target site. For example, if you are having a liver problem and you get a hepatotoxic or hepatopreventive agent, it does not 100% reaches to your liver. It gets into your GIT. If you are having it from oral route, it gets absorbed, distributed, and when it reaches systemic circulation, that is your blood circulation, the drug gets distributed throughout your body, and only a fixed proportion of the drug will reach to your disease cells. So here in phase 0, we calculated how much amount of a drug is actually reaching the disease cells. What happens to the drug in body, as many of the drugs are having metabolism or some destruction because of the enzymes that are present in the body. How body cells respond to a drug, because every drug is having different response from different anatomies and physiologies. We perform blood tests and disease tissue biopsies to check out this. Next slide. These are the advantages and limitations of phase 0 trials. As I have told you, there is nothing new. It is about that we have to check whether the drug which we are claiming to have effects in humans, either it is capable of that much potency or not, because we cannot compromise human subject at any cost. Human safety and subject safety is a very major cause whenever we are doing clinical trials. Next slide. The phase 1 trials. It is the first stage of testing in human population and is designed to assess safety, tolerability, pharmacokinetic, and pharmacodynamics. The difference between pharmacokinetic and pharmacodynamics is pharmacokinetics includes ADME, that is absorption, metabolism, distribution, and excretion of the drug. While pharmacodynamics, it includes that what your body does to the drugs. Includes smaller number of subjects ranging between 20 to 25. Phase 1 trials include only healthy volunteers. Here we are not going to include any kind of disease volunteers, because here we are checking safety and tolerability of the drug. So it includes only 20 to 25 subjects in phase 1. Next slide. The aim of the study is to determine the maximum tolerated dose. Human body is very, very sensitive. We cannot tolerate high doses, or even sometimes when we are having a potent drug, even 1 or 2 milligram of a drug can prove fatal to human body. That's why in phase 1, we use healthy volunteers and then we check for the maximum tolerated dose. It is found by the escalating treatment doses, and it is also called as dose escalation studies. Here, the first 2 patients that take part are given very small dose of the drug. If it goes well, then we gradually increase the dose up to which toxicity does not occur, because we are aiming to find out the maximum tolerated dose. And here we find out the best proposed dose that is administered to a patient or a healthy volunteer to cure the disease. Next slide. There are two kinds of phase 1 studies. One is single ascending dose and other is multiple ascending dose. Here we also do food effect studies. That is, whenever we have a drug, either even some of you have heard that some drugs are advised to be taken empty stomach, some are advised to be taken after meals. That is where we investigate the effect of absorption pattern with food. Some drugs are absorbed better when the stomach is filled with meals, some drugs go better when the stomach is empty. Each and every drug is own characteristics, and that's what we have to find out. Even we have to study the food and drug interaction, because you might have heard that tetracyclines are advised to not to be taken with milk. The reason behind that is that milk, which is having calcium ions, caused chelation, and tetracycline got chelated, and the effect of that antibiotic in your body get nullified. In phase 1 trial, the volunteer receives no therapeutic benefit because they are healthy volunteers, they are not having any disease, so they are not having any kind of therapeutic benefit, and that's why it is called non-therapeutic research. The phase 1 studies only. Next slide. As per the regulatory bodies, authorities, either US FDA, CDSCO, or whatever country it is having, every country is having their own regulatory bodies, and every regulatory body have requirements to include the special population, which is women of childbearing age, because in human physiology also, females and males are having very, very different responses against the same molecule in the same dose. Children, if required, if the drug which we are planning, the molecule or the intervention that we are aiming is for children, then we have to test it in children also. It is also called pediatric if it is including very, very small children. And then geriatrics. The patient above the age of 60 are considered under the geriatric category of the patients. It is called as a special population because these three react differently as the way a normal adult will react to a considerable dose. Next slide. The phase 2 trials. The phase 2 trials are also called as therapeutic exploratory trials because here we explore the therapeutic efficacy of our drugs. These are very larger in comparison to phase 1 because here we are including 25 to 300 subjects. The number of population is increased in this. And in phase 2 trials, the new treatments are compared with the standards as well as placebos. Placebo is something that a drug which actually mimic your original drug. For example, if you see a paracetamol placebo and a normal paracetamol that is available in market, you will not be able to check whether which one is actually a placebo and which one is not. And placebos are not permitted to be manufactured without concern of DCGI. A special batch of a specified number of drugs are only permitted when we are having trials, and then only we are having the placebos being manufactured. Otherwise, they may lead to severe fraud with public because placebos are no way differentiable with the original drug. They are made exactly the same way, packed exactly as the same way a marketed drug is packed, and you will not be able to differentiate that what a placebo is. Only the one who is sponsoring the trials is able to know, and if he or she requests that they need to compare their molecular intervention with a placebo, then the DCGI permits them to have the limited amount of placebos to be manufactured. These are more randomized, and that means that number of people are in groups are random in these kind of trials. And even in last few days, we are having some of experts with us and they told us that the randomized kind of trials are the most appreciated and acceptable kind of trials that give us promising results. Next slide. The aim of phase 2 trials is to check out the efficacy of the drug, to evaluate its safety, and to monitor its side effects. And the most important, the optimum dose finding. It is done by dose efficacy relationships, therapeutic dosage regimen, the duration of the therapy, the frequency of administration, and the therapeutic window. These are the governing factors for determining an optimum dose finding. Next slide. The volunteers in phase 2 are the patients. In phase 1, we are having the healthy volunteers, but in phase 2 trials, we are having the diseased or the patient kind of volunteers that are suffering from the disease of which the drug is expected to treat. For example, if we are having an anti-neoplastic drug that will be used for the cancer patients, then we will include cancer patients in phase 2 trials. If we are having any anti-diabetic drug, then we will include diabetic patients. The kind of patients are decided according to the action of drug that we are claiming to be having. May take up time up to several years. These are way long trials and take a long sort of time. Phase 2 trials are of two types, phase 2A and phase 2B. Phase 2A is more prone to the decision of the dosage requirement, that how much amount of dose is required to have the therapeutic effect. While phase 2 trials, 2B trials are designed to study the efficacy. Efficacy is determined by the minimum dose with maximum effect. Next. These are the various comparisons between the phase 2A and 2B slides, which I told you yet, that the new thing in this is that single blind and double blind. Whenever we have trials, a patient is not always told that what he or she is being administered. And in double blind trials, even the healthcare professionals involved in the trial, they are also unaware that what kind of molecule they are using in trials, what is the molecule that they are administering to their patients. Next slide. Next slide. Next. This one, phase 3 trials. So in phase 2, we have determined the efficacy and the safety of the drug. And here comes the most crucial phase of the clinical trials, that is phase 3. It is confirmatory trial. It is a large-scale, multi-centric, randomized and controlled trial. Here the target population ranges from several hundred to 3,000 patients. It includes a very large number of subjects and study maximum possible side effects and collect safety data. Here, the determination of the therapeutic dose occurs with the help of therapeutic index. Therapeutic index is defined as the ratio between the lethal and effective dose, that is ED50 upon LD50. ED is the effective dose that causes effective and efficient cure to the 50% of the population of the test group, and LD50 is determined as the dose that causes lethality or harmful effects on 50% of the test group. Assessment of relative therapeutic value. The relative therapeutic value means the safety, efficacy, and if some special properties that exist in our molecule or intervention. Next slide. Phase 3 trials are also of two kinds. Phase 3A, it is done before NDA approval is applied for obtaining the sufficient and significant data that will promise that your molecule or intervention is beneficial for the human subjects. And phase 3B, it is also called as label expansion studies, and it is done not prior to NDA but before the drug is being launched in market. Because when you apply for NDA, it takes several years or up to 7 to 10 years for NDA review, and then only after a lot of experimental proofs, review, and editing and omission, your drug will get approval, and if it is getting approved, then only it will go for phase 4 trials, that is marketing. The phase 3B trials are done to check whether the drug is having any additional kind of effect beyond the original use for which it is being aimed. For example, we are having a drug for antihypertensive effect, that is, it is lowering your blood pressure, but in a long run or in a long use, it may cause some harmful effect or some beneficial effect also, but that will take a very long time to be able to be visible. Not all effects of drug are visible. The drugs which cause accumulation can result in even 10, 15 or 20 years after trials, and they are then withdrawal from the market. Next slide. So here we are run with the phase 1, 2, and 3 of the clinical trials. In phase 1, we use healthy volunteers. In phase 2, we use disease patients. And in phase 3, we use a large number of disease patients to obtain the safety, efficacy, and potency of the data. After the phase 1, 2, and 3 promising results, we apply for the NDA. The NDA is filed with the DCGI. The NDA filing includes expert committee review for efficacy, safety, and potential sales. That is, we have expert committee from the sponsors, they review for whatever the results we are having from phase 1, 2, and 3 trials. And then only when they found it promising and beneficial, they apply for the NDA application. When the NDA is applied, the DCGI, that is the Drug Controller General of India, he is having a specified expert review committee. They review your drug, your molecule, your results. And if they found it promising for human subjects, then only they will approve you. And if you get the DCGI approval from NDA, your drug is being marketed and the beginning of phase 4 trials occur from this point. Next slide. The phase 4 trials that are done after the NDA approval. In phase 4 trials, the drug that has been shown to work and has not been licensed, these kind of drugs are approved and marketed, but to whomsoever they are marketed, the data is always maintained. The record of the sales is maintained very well, so that if we have to withdraw the drug or we are having some safety indications, we will be able to involve and inform our consumers about the harms or the benefits. The time and duration of this phase 4 trials has not been fixed. For example, the population size for phase 1, 2, and 3 is fixed. But for phase 4, it can reach up to several thousand of patients. These kind of trials inquire more about the side effects and safety of the drug in long run. What are the long-term risks and benefits? How well the drug works when it is used widely and on various kind of population and conditions. These are also called as post-marketing surveillance. That is, when the drug is being marketed, even after that, you have to maintain the record of the data obtained from the people who are using it. And these are not experimental. In phase 1, 2, and 3, we are having experiments on human subjects. But in phase 4, these are observational kind of studies where you do observe whatever the results are being obtained from your trials. Next slide. Here, the study of ADR, that is adverse effects, is done, and the drug interactions, which comes under the pharmacovigilance. Pharmacovigilance is a stream of science that deals with the ADR, their monitoring, and their control. It helps us to explore new unpredicted uses of drugs. For example, minoxidil is initially used as an antihypertensive, but the novel use, which is the side effect earlier, that it causes hair regrowth, is now being used as the major use of the drug, and minoxidil is being used in alopecia. Periodic safety update reports are always maintained up to almost 4 years post-approval. That is initially every 6 months. The reporting of ADR, that is adverse reactions. No, after it's fine. It is an example that in September 2004, a company withdrew its drug from market due to increased risk of heart attack and stroke in long-term use and high usage of the drug. When a drug is having serious toxic or hazardous effect, even after a long term after being marketed, the drug gets withdrawal from the market. For example, you might have heard about the market withdrawal of Zentic products, that is ranitidine, which is having NMDA, which is a potent carcinogen. Next slide. Reporting of ADRs. Adverse drug reactions are then unintended, that is, we do not intend for this effect, and noxious, that is, harmful kind of effect that is being observed at the normal therapeutic dose. Here, the word normal therapeutic dose is very important because therapeutic dose is the amount of drug that is needed for having a pharmacological effect. And when at that prior dose, you obtain some unintended or noxious effect that is called as ADRs. It is meant to be reported essentially, and WHO have a separate headquarters to handle these issues. The UMC, that is Uppsala Monitoring Center, situated in Sweden. It is the global center for pharmacovigilance and adverse drug reaction monitoring. In India, we are having Pharmacovigilance Program of India, that is PVPI, and it is governed by IPC, that is Indian Pharmacopoeia Commission, situated in Ghaziabad. Next slide. So here we conclude that clinical trials are human experiments. Preclinical trials are animal experiments. They are designed to study safety, efficacy of the new drug or intervention. It includes 4 phases with specified objectives and results. Here we are having 2 kinds of applications. That is, one is IND, it is filed and it is having a review time of 30 days. Either in 30 days your drug get approval or you will be requested for additional information. But in India, it takes up to several years to have a drug approved. Well-designed and executed trials for the base for therapeutic decision. They must follow all the ethical guidelines and protocols. Clinical trials are done in relevance to all the ethical guidelines and protocols from the governing bodies of the countries as well as the global bodies. Next slide. So here it is all about clinical trials that I was about to share. Next slide, please. Here I have quoted something that there are 3 kinds of actions of a drug. The one we want, the intended effect for which we are using a drug, the one we don't want, that is unintended, undesired effect, and the one that we don't even know about. So because several kinds of effects of a drug are remaining unexplored, uninformed up to several years of use. So that's all about it. Now, any one of you is having any query, I'll be very glad to answer it if I possible. It's done from my side, doctor.
Thank you, Kirti. The session is open for discussion. If you have any doubts, you can ask Kirti. Any questions?
Hello.
Yes, you are audible.
I had a question.
Sure.
First of all, a very good presentation.
Thank you so much.
Generally, not only for the drugs, but also for vaccine, we have the same clinical trials, right? So generally, if it is a vaccine or a drug, it goes through all these clinical trials. But when we have an emergency situation, it does not go through all the clinical trials, right?
Yes. There we are having a protocol of emergency use authorization.
Correct. So in that case, how will you categorize the drug as safe without it going through all the clinical trials?
Generally, this process is of a very long tenure that we have mentioned. It is having almost 25 to 30 years for a molecular vaccine to be proven safe for human usage. But as likewise we saw in the case of in condition of COVID, the EUA, that is Emergency Use Authorization, it is provided only in the conditions when the drug is having primarily safety data in preclinical and human trials in phase 1 and 2. Here we have accelerated the process and the authorization is allowed only when the drug or the vaccine claims and is being able to prevent the number of deaths that are being caused. For example, in COVID situation, we are having a large number of fatal cases before the intervention of vaccine in our trials. So we have to give it an emergency use authorization. US FDA is having several guidelines. If you can check it up on US FDA portal, they are having several set of guidelines in which conditions you can approve a drug for emergency use authorization. I hope I have answered it up to my level of knowledge.
Yes. Thank you.
You're welcome. Anyone else?
There is a question in chat box.
session is good in the case the drug fails at phase 4 trials, what will be the immediate remedy we can take to clear phase 4? Initially, phase 4 is done after a large set of reviews. So the cases of such harm or such serious side effects is very low. And even if something occurs like this, then the remedy is that the company withdraws a drug from the market. That the examples I have quoted, and even after several years of safe use. For example, ranitidine is being used in human race for a very long period. It is considered safe and it is an OTC drug, that is over-the-counter drug. You can buy it without prescription. But because of the presence of NMDA, that is present in your food and water also, but in up to a limit, which is not harmful or carcinogenic to your body. But having a daily regular usage of ranitidine, and if you are using it consequently, the NMDA has proved to be a very potent carcinogen, and that's why Zentech has withdrawn its product from the market. And even in several countries have banned the use of such kind of drugs. Market withdrawal of drug is done, and that's why we are having the UNC, the Pharmacovigilance program, which is having the regular monitoring of ADRs. And you can report, any one of you who is using a drug can report the ADRs.
Any other questions? If no more questions.
If a drug fails in any level, does it have to go through all process from the phase 0? Whenever a drug fails some process, it depends on the level and intricacy of the failure. For example, if it is having some failure because of some minor effects, then it is started from the phase up to which it is successful. For example, a drug is being successful up to phase 2, but in phase 3, it suddenly gives us non-promising results. Then we started again from the phase 2 trials. And we check out the written protocols, the procedures, the standard procedures, that because there are a number of errors that are possible. Either it can be a clinical error, an instrumentation error. Even in case of drugs, we have errors due to the availability of dosage form, the routes, the time of administration, the frequency of administration. Everything is having its own individual effects. For example, if you give a drug from oral route and intravenous route, the bioavailability, the kinetics of its absorption, distribution, metabolism, everything is very, very different. And even if our drugs have hazardous or non-promising effects in phase 3, either it has to start from phase 0, or sometimes the drugs are being rejected totally if the molecule is being hazardous or fatal.
Thank you.
In case I'm designing a drug similar to existing drug, you change a few properties. Can I make direct phase 3 trials? No, you can't go for phase 3 trial directly, but you have to start from phase 2 trials. For existing kind of drugs, if you are having a comparative study, you have to start from phase 2 trials in which you involve the diseased volunteers. And you have to compare your set of data with that of the standard marketed drug present. Any more questions?
Any more questions?
I hope I had somewhat up to.
It was a good presentation, Kirti. Thank you so much
Professor.
for giving us an overview of clinical trials. Hope all students have understood some basic concepts of clinical trials from your presentation. Thank you so much.
Thank you so much, Professor.
Students, we will end up our session for the day today. The questions for quiz and assignments will be shared in the WhatsApp group. Okay. Thank you so much for joining us today.